Good afternoon. It's my pleasure to introduce today's keynote speaker, Professor Fernando Lopes Da Silva. Professor Lopes Da Silva is a world-leading figure in electrophysiology and biophysics of brain systems. His seminal work on the basic mechanisms of cerebral rhythmic activities led to the discovery of the cortical origin of the algorithm of the brain, a discovery with major impact in the understanding of attention, of consciousness, and of sleep. Likewise, his work in epilepsy had major impact in the understanding of this pathology. His discovery of the network responsible for the initiation of absence epileptic seizures, what he called the cortical focus theory, has fundamentally changed the view on the pathophysiology of this disease. Professor Lopes Da Silva was the first director of the Institute of Neurobiology of the Faculty of Science of the University of Amsterdam, where he is still active as emeritus professor now. While he spent most of his academic and clinical life in Amsterdam, his Portuguese roots led to tight links with Lisbon, where he is now professor at the Faculty of Medicine of the university, as well the Technical University of Lisbon. Professor Lopes Da Silva is member of the Netherlands Royal Academy of Arts and Sciences. He received degrees of Doctor Honoris Causa from the University of Lisbon, from the University of Porto, and from the University of Helsinki. He received the Herbert H. Jasper Award of the American Clinical Neurophysiology Society, degree of Grand Officer of the Order of Santiago de Espado of Portugal, and finally the degree of Knight of the Order of the Netherlands Leeuw, the Dutch Lion. Professor Lopes Da Silva has published many highly cited research articles, review papers, and book chapters. Most important, his role as leading editor of what we like to call the Bible for all clinical neurophysiologists, the Niedermeyer's Handbook of Electroencephalography, a book with over 1,300 pages, which is soon publishing his sixth edition. So please welcome with me Professor Lopes Da Silva for this keynote lecture.
So thank you very much, Christoph, for these nice words. I hope you can hear me, okay? And also, for inviting me to deliver this lecture, which is, of course, an honor and a pleasure for me. Actually, the edition is not the 6th, it's the 7th, Christoph. Oh. I always, I am accustomed to talk with students, so we have. Anyway, I decided here to talk in a way rather, let's say, simple, just to keep the words of Albert Einstein, who always said, that you should give talks that are really simple, very simple, very, very simple, but not simpler. So I hope it is not simplistic anyway, but still, to focus on a number of items that are, I think, of interest from the point of view of electrophysiology in the brain and the ideas we have about this at this moment. If I have the next slide. So my outline of this lecture is that I start with some very short comment on measures of functional connectivity and then go into measures of causal connectivity with emphasis on the concept of transfer function and concept of phase and time delays. And then I'll take the topic, which is very common today, of Granger causality and different methods used to estimate Granger causality. And then at the end, I will go from the autoregressive filter approach that is very much used in causality studies to neural mass models, testing specific hypotheses. So I start with the question of functional connectivity. There are many methods now that are in the literature that many people here will be familiar with that are being used to study functional connectivity. These are methods basically using correlation in one way or another. They are being used with low focal potentials in the brain. They are being used in EEG, in the ECG, in MEG, very similar methods. But the question that is put very often in practice is which method should one use? And I think that in this respect, we don't have a very simple answer. However, I think it's very important, and I'll come back to that at the end of this talk again, that people do a comparison of the performance of different methods using the same methodology and without bias. Because, of course, I mean, some people who publish one new method usually are implicitly defending that method as being the best one. But you have to test this in an unbiased way in different kinds of situations. So you have to use simulations that are really good ones, and they can be used then to evaluate which is the performance of the methods. It's not the question of knowing which is the best, but the question is of knowing which is the best for certain purpose. So the indications of the different methods. In this paper that I mentioned here in this slide of Fabrice Wendling, it was published in 2009. I mean, what they did was exactly, I mean, to simulate different kinds of EEG, MEG kind of signals with specific properties, very similar to situations that you find in practice, and using then a method of comparison of these outputs of these different sources. Source number one giving one particular signal, source number two, which is on the right, the other on the left, giving another particular source of signals, and then modifying the coupling constant. I mean, that relates one to the other, either in amplitude or in phase and amplitude, and then applying to the result, after adding noise, applying the different methods that are available. And these methods are listed on this list on the left. Anyway, I'm not going to go into details about these methods. You can read this in this paper. It's very well done. But I'd like just to draw some conclusions. And the conclusions that can be drawn from this is that some methods are poor sensitive to coupling parameters. So, I mean, you'll be aware of that. Second one is, in general, methods performed better to broadband signals than to narrowband signals, which is not surprising. I mean, because broadband signals are more noisy in a sense. I mean, so they are more easier to get correlations that are significant. There is no ideal method. I would say forget it. There is no ideal method. However, it is important and it is recommended to compare results of different methods and try to that in that way to get some reliability of the methods or the results that you are obtaining. And fourth, I would say that regression methods, simple regression methods, linear or nonlinear regression methods, are recommended to be used as a sort of first analysis. They are very simple. They can give you some indication about what is important in the functional connectivity between different kinds of signals, the correlation of these signals, and they can be then used again to compare the results with more, let's say, sophisticated methods. So this is rather simple in the sense that what we now can do with this information is that we can get a better idea of what is the choice of the method that you can use for functional connectivity. As I said, this next step is to talk a little bit about the next stage. And the next stage is to introduce to you this very simplified idea about causality. So what I start here is from example number one, where we have a signal, which is a noisy signal, which as input. Then you have a transfer function. You have a transfer function with a certain time delay, and you get an output signal. Okay? So what is the situation here is what you can have in the brain if you have some activity going on in one particular part of the brain, and that projects by means of anatomical pathways to another part of the brain. So you want to determine the relationship of the causal relationship between these two. What is most important? So I'll try to go with this very, very simplified model a little bit further. So I'll show you some data that were obtained in animal experiments, but of course it could be also done in human. In this case, we are looking at results obtained by recording activities in the limbic cortex. So we have activity in the limbic cortex in the so-called PPC, which is the prepiriform cortex, and you have recordings done simultaneously in the entorhinal cortex. We show here just 1 second epoch of both. You do a Fourier analysis, which is simple power spectrum analysis, and by using this analysis, you can determine something about what I think is the point I would like to stress is the importance of the error of time. Granger himself said, I mean, one of the most important things in causality is not only the direction of the relationship between signal A and signal B, but it's also the time in between. And that's I'd like to try to show it to you. So here you have the spectra of both signals, and as you see, the spectra of the PPC is a bit different from the one on the entorhinal cortex. PPC has a very strong activity in the beta-gamma oscillations. The same applies to the entorhinal cortex, but it is less pronounced. And what we can do is to do a cross-power spectrum between these two signals. And by viewing the cross-power spectrum, you can deduce, I'm not going to the methodology in detail, you can deduce what is the phase relationship between both. And here you have the phase plotted along the vertical axis against frequency plotted along the horizontal axis. And for the frequency band that is given in black under the frequency and the horizontal axis, if you look now to the phase, you see that the phase is rather close to a straight line. So that is an important result. And that gives you the possibility of deducing from this a time delay. So what you have to do now here is by using an analytical procedure, using the Hilbert transform. As I said, I'm not going into details about the techniques. You can determine what is the phase that corresponds the best, I mean, to this particular situation, and you can determine the phase slope. And from the phase slope, you can determine the time delay. So this simple equation, delta t is delta phi divided by delta f. This gives you the slope, and if the slope is indeed a linear slope, this gives you a robust estimate of the time delay between the two signals. Sometimes in some methods that are being used, it looks like everything has no time explicitly mentioned in the analysis of relationship between signals. But everything in the brain takes time. So time is absolutely essential. I would like just to stress very much this point. So is this experiment, I mean, it's not very important now, but it could be determined with quite exactly that we had a time, let's say, of 9.3 milliseconds, and this corresponds exactly to what you would find if you stimulate one area and record from the other area, really. This is now, okay, the spontaneous activity, but the evoked activity fits exactly this, and you can deduce from this propagation velocity, and you can see if that is compatible with the diameter of the fibers. So I'm just insisting on this because this is, of course, rather basic, but still it gives you the idea that in essential correlation in the terms of causality has to do with transfer functions, and has to do with phase, and has to do with time delay. So that is my, message here. A second example is taken from work that is recently published by Timo van Kerkhove. He is working also in Amsterdam, in the Neuroscience Institute in Amsterdam. And it's quite different. They work in the monkey, and in the monkey they were recording activity from the visual cortex. From, if you look at plot A, you see the V1 and V4. So they were looking at the different communication and causality, actually, between V1 and V4, the primary and other areas of visual cortex. So the interesting result here is shown particularly in C. If you look now, I focus now in C, and you see now they plotted against frequency the phase. And you see this for different frequency bands. So if you look carefully, and you look first starting from the zero phase to zero frequency, say, you see a line with a slope going from up left to down right, which means that you have a time delay going from V4 to V1. And it means that it is a time delay for the activity in the alpha and the lower alpha range of frequencies. On the other hand, if you look to the right side, what you see is a rather less simple line, but still a slope going into the opposite direction, going from lower left to upper right. And this slope corresponds to a time delay of only about 3 milliseconds. And this one here is in different direction to the previous one, indicating that we are looking now to the correlation of activity, the causal relationship between V1 and V4. So the conclusion of this is that you have between V1 and V4, I think this is a very interesting, important conclusion. You have two directions and two frequency bands where the activity from V1 to V4 is mainly on the basis of gamma activity. You see the spectrum is all on the high frequency band, and it is very quick. And then you have backwards from V4 to V1 in the alpha and lower frequency band activity that propagates a little bit slower. So it is an indication that you have indeed feed-forward information and feedback information, and the feed-forward and feedback channels of information use different oscillations, different frequency oscillations, and they have also different conductivity velocities. Now, this is a wake monkey. So if you would do this now in the cortex, the surface of the cortex, you can find, of course, this effect. Probably if you would record this from the EEG or the scalp EEG, it will not be so easy to get to this to distinguish these the two cases. But still, it's very important. I mean that we refer to this kind of information, which is basic information in terms of the processing of information in the cortex. In this case, in the visual cortex. So now I'd like to go a little bit more to the Granger causality applied now in different kinds of levels, from the electrocorticogram to the scalp, and in this way, starting from the autoregressive modeling, which is the basis of Granger, and many people speak about Granger, but I don't know if many people have read Granger. If they have not, they should do, because it's rather entertaining. You know, it's also entertaining to read people who really made something fundamental contributions, talking about their own contributions. And in this paper, so testing for causality, a personal viewpoint, that's exactly what I mean, is published in the Journal of Economic Dynamics and Control. I think it's a journal that most neuroscientists don't read, except, I mean, if they are interested in the financial affairs, which some are, of course. But anyway, I don't know if they will get very far with Granger on that respect. But the definition is given by himself, so this is a quotation. The definition of causation essentially says that you have a signal X, from T and J, will consist of a part that can be explained by some proper information set of the whole signal, plus an unexplained part. Now, if signal Y can be used to partly forecast the unexplained part, then Y is said to be prima facie cause of X. So that in this way, in fact, you reduce, let's say, the uncertainty about signal X if you add the information from signal Y. This is the beauty of Granger's approach is simplicity. So it goes in the direction of what I said about Einstein in the beginning. It's very simple, although this sentence is perhaps not the simplest English sentence that I have met until now. Anyway, some of methods that have been now developed, I mean, to implant Granger causality, again, there are several methods. And I'm not going to give a list of methods and the description in detail of methods. I just pick up a few examples. This one, I think, is interesting as an illustration. Is the so-called directed transfer function. And that is actually the short-term directed transfer function. So it's in fact a way of having an approximation of the transfer function between two signals recorded from two sides in the brain. And it is directed, and this meaning that you are looking at the relationship directly, I mean, to this of one signal causing the other. And it is the word direct in the beginning means that by using partial coherence, you can eliminate the relationships that are intermediary between A and B and C, etc. So you go only to the ones that are really the most direct ones, and the rest can be eliminated in this way. Now, this is an experiment done in Johns Hopkins by Anna Korzenikowska, and she's from Poland, and in fact, the Polish group in Warsaw has done quite a lot in developing this type of techniques and methods, and has been applied to electrocorticograms obtained from epileptic patients in the group of Nat and Krohn in Johns Hopkins. This task, in this case, is an auditory task. So you see there are 3 periods in this task. The first period, on the left, shows you the auditory perception stage. Then you have the auditory preparation to the response to the auditory stimulus. And third, you have then the verbal response. So if you look now carefully to the first one, A, you see a lot of yellow lines. Yellow lines have always a direction because, of course, this directed transfer function gives you direction. It's a symmetry in this case. But some are more important than others. Most important ones are given in red. And as you see from in the left, you see that there is a flow of information from auditory association cortex, which is the E9 electrode mainly, up to the area in the motor cortex of E4, E3, which corresponds to the mouth and tongue motor cortex. So this is actually in the auditory perception time. So we're going information immediately, say, from auditory cortex to areas that are going to be important for the response. Then in the second stage, the response preparation, you see a lot of information going on in parallel. And if you look carefully, I mean, you see that it goes in different directions, and that there is a lot of parallel processing going on. But there are 2 areas that are most important ones. One, we have the red arrow again. This is an area between the auditory cortex and Wernicke cortex, going a little bit more to the right side in this plot, and projecting again to the motor cortex. But you see also this activity going in both directions. You see two arrows going, one in and the other one in the opposite directions, which are related to the auditory cortex and Broca's area. Broca's area being the one more in the between, say, electrode E2. And so that this way you can see that during this preparation you have at the same time some information that is being processed in the Wernicke auditory part, and at the same time information that is going on already in the Broca's area. You don't see very much connection between both directly. These are going in parallel. And in the third, you see the response, and that is quite clear. There's a very strong association between from the Broca's area, more anterior area, to the motor cortex, where the mouth and tongue are represented. So you see in this way that by using transfer function in this way, you can indeed determine the sequence of events, always keeping very clear the relationship in the direction of the information flow from which area to which area. What is missing here, I must say, is really time. you have the arrow of time in the sense that you have the direction that information is going from A to B, but you don't have a precise information about time. That is something that is important to add to this kind of methods. And some people are working on that. So I'd like to go to a second example. And this second example is taken, from work done here in Geneva. And it gives me the possibility of touching another aspect which is important. And this is the question of whether you can use information from the scalp instead of using information only from the electrocorticogram or intracerebral information for this kind of studies of causality. And this is a nice example that you can. Of course, you have to be aware that to do this, you have to go, from the scalp activity to the sources in the cortex. So I have to say a few things about that. There's a lot of people doing this kind of studies. This case is a study done here by Anne Coito in the group of Christophe Michel, and she defended her PhD thesis last week. And I had the privilege of being also examiner of this thesis, so I have to read it. Yes, I did. Anyway, the point is that the question was a question for the following situation. These patients are epileptic patients, and they are epileptic patients that have epileptiform spikes. They have interictal spikes. Sometimes they don't have, but they have some periods where they have these interictal spikes. So the question was, can you say something about the sources of the epileptic activity taken from these epileptiform spikes? So in this case, you take the epileptiform spikes from the scalp, and you do an inverse solution, which is, of course, always an approximation, okay? So by using a dipole kind of inverse solution, you can determine the most likely sources at the level of the cortex. Now I have now to say something which I think is maybe very simple again, but let's keep on with Albert Einstein. It's not simpler, and that is the following one: is that what is a source then? Let's think about this. In the brain, there is some generators of epileptiform spikes in the brain, in the cortex, in several layers of the cortex. And they, because of biophysics, they are possible to be recorded from the surface, from the scalp. And in the MEG, they even be outside the scalp. They can be censored by the MEG sensors. That's okay. But in doing this is a forward problem, you lose information. It's inevitable. You lose information because of the different tissues between the cortex and the surface. Then, with this this information that is already lost, you do inverse problem. So you have to do this by some approximations, which you need to have some regularization algorithms, which of course are important and can be different ones. And in this way, you get then back to an estimated source at the cortex. But don't think that the estimated course is the same as the original course. It's not the same source. It's just a picture of the source, but it's not the same one. So I'd like to stress this, just to say that sometimes people think that you talk about sources, and source is just one name, and it can be applied to anything. But it's not exactly the same thing. One is a source that is really the source of the activity in the cortex. The other one is an estimated source that is due to this forward inverse approximation. Anyway, to go back to this analysis, what she did here, and Coito applied another method of causality, which is the partial directed coherence, which is very similar to the transfer function, I must say, but it has some specificities. And in the way, I'm not going into detail of that, because it takes into account not only, let's say, two electrodes, two sides, but takes into account all the others. So it takes into account, how much flow is going from all the different sites that are recorded up to a particular site that is now considered to be the reference. So it takes all this into account. This is why that is in the denominator of this formula. Now, this way, the partial directed coherence delivers, of course, then information about outflows of information from one particular site to all the other sites. And this is now represented by means of the circles, which you see on the red, and some are blue and some are red. In this case, there were two groups of patients. There were patients that had epileptic focus on the right side of the brain, so that is RTLE, temporal lobe epilepsy. Others had focus on the left side. If you look at the right side ones first, which are on the right, by chance, this you see a red circle with a rather big circle on the most anterior part of the area corresponding to the hippocampus. And you can also see that there is a projection with that is causal relationship between the right hub, let's say, of activity going into the left side. So these lines are yellow, and so they have a direction from right to left. On the other hand, if you look at the left case, on the left, of course, then you see the distribution is a little bit different. You have two sides, I mean, one more anterior, the other more medial. You have a lot of communication between both. You have a lot of lines with arrows indicating communication between anterior and posterior focus. But you have, surprising enough, because that was not known when before this was done, I think, you have surprisingly very little communication between let's say, the left side and the right side. So there's a big asymmetry in this. The right side engage a lot of, entrain a lot of activities in the contralateral side, and that is not the case in the left side. Also interesting, but I don't have any possibility to go into detail on that, is that these patients had also a different kind of cognitive deficit. Cognitive deficit in the cases of the patients that had the right temporal lobe epilepsy, different from the left temporal lobe epilepsy, and this has to do also with the fact that the entrainment was very strong in the case of the right side, but not in the case of the left side. So just to give you an idea that you can go, I mean, with this methodology in the brain, you can study in the brain like in the monkey. You can study this at the level of the cortex, electrocorticogram, but you can also do it at the level of the scalp. So another method that is now becoming even more popular is the transfer entropy. And transfer entropy, I don't go into any detail about the methodology here. The transfer entropy is, in fact, a method that is derived from the very general idea, and already for a long time known in statistics. It is called mutual information. So mutual information between two signals, conditional on a third signal. That is the basic idea of transfer. There are several good articles on this, which I just mentioned here, the one from Roux and the collaborators in Journal Neuroscience, and the other one from Lobler and the collaborators in Neuroimage, very recent ones, 2013 and 14. And I take one example from this Roux study, which I think is very interesting. But before that, I have to say that we're talking about different methods, but if you are interested really in the mathematics behind all these different methods, the directed transfer function, the direct transfer function, this transfer entropy, etc., PDC, then you see that most of these methods are equivalent. For Gaussian variables are equivalent. And they are different in the implementation, but they are not different in the basic mathematics behind them. So I said I'd like to show this example from the work of Roux and others, where they did a study on MEG. So from MEG, now we come now for MEG, where they're using filtering techniques, so beamforming kind of techniques. They localized activities in the gamma frequency range, which is shown in the pictures A, B, C, the coronal and horizontal sections of the brain. These are the gamma, say, hubs of activity. And there is also activity in the alpha that's not shown in this picture here. And the question they wanted to answer was how much of this gamma activity, power in the gamma activity, is modulated by the phase of alpha activity. Alpha activity that was recorded particularly in this case from the thalamus. And I'd like to jump to one aspect of this analysis, which is here on the right. They were able to convincingly to show evidence that in fact alpha activity recorded in this way, using this beam-forming technique in the thalamus, was modulating gamma amplitude. And they found also that this alpha activity was propagated, from thalamus to cortex with a certain delay, and they could measure the delay, which I think is really a very interesting addition to what has been done with other methodologies. And this is what is shown in this square, this red square, where you see the probability distribution of the delays estimated from the thalamus to the cortex. And you see the delay, the mean delay or the mode of this distribution is about 15 milliseconds. So when I say this, I could not help to think about something that I did many years ago in animal, where we were asking exactly the same type of question. We are asking about how much of alpha activity in the thalamus is projected to the cortex, and how is that being done. So we had electrodes implanted in animals with different sites. That some numbers are given there, but we had many more electrodes, but some of them in the marginal gyrus, some of them on the calcarine area. So this electrodes were 11, 10, 9, 8, etc. And we had electrodes also in different nuclei of the thalamus. Here in this case in the LGN, electrogenucleic nucleus, and also in the pulvinar. And now we found, by using, in this case, Fourier analysis, using phase analysis of Fourier analysis, we found that indeed that activity going from the genetic nucleus was projecting to some areas of the cortex, and we were able to determine the time delay. And this was in the order of 13 milliseconds. And the pulvinar was also projecting to some other parts of the brain with a different distribution. I'm not going into detail on that. And they were something like about 17 milliseconds. So I was really astonished when I saw this work done with MEG using these techniques in human, that we are very close to the same kind of time delays, although there's animal and human, there are differences and so on, but still it's very compatible, one to the other. So that is one thing I'd like to tell about. And I would like to stress still one other thing in this. If you look to the graphs, to the plots on the right side, you see, in fact, a lot of curves which show the coherence between different electrodes with different sites in the cortex. Let's say take the first one the first one. So you have between 15 and 10, and you have around 10 hertz, you have a peak of the coherence. This is a coherence function, okay, linear coherence function. But now the question is, where is this coherence reflecting, in fact, information flowing from one to the other electrode or electrode site, or is that depending on some other variable? So we did partial coherence, the partial coherence also was done in that research, that study done by Korzeniowska. And in fact, what you see now is a demonstration of the effect of what Granger has called the missing variable. So when you do any correlation, causal correlation between any pair of signals, you have to know the whole set. You have to know, are you missing something? And in fact, when you did the partialization, now you look to the third trace. And the third trace, the peak in the coherence between the two electrodes of the cortex, there are two electrodes in the cortex. Now we're partializing this to the thalamus, and it disappears. This was actually the demonstration that these different areas of the cortex are receiving divergent inputs from the thalamus. And in fact, we didn't know that before, and afterwards this was confirmed really very well in anatomy. Okay, so just to finalize, I go a little bit to the question of this modeling. And I like to say that it's important, to tell you, I think, that many of the models that are now being used, so in the so-called dynamic causal models, particularly the group of Carl Friston and others have done a lot of work on this, are really derived from models of neural mass models that were developed in the 70s. And in this way, I'd like to make a small intermezzo here, historical intermezzo, just to say that all starts, I mean, from the very beginning, from Wilson and Cowan model that was published in 1972 for the first time and then 73. And people who are interested again in the history of this could read a very interesting paper from the Steks and Sainovsky that is indicated there in under and below in the slide that was published in Biological Cybernetics, the Wilson-Cowan model 36 years later. Now it will be a little bit longer than that. What is now the basics of this? The basics is that instead of trying to make just mathematical models simple, no, we go into neurophysiological responsible models using neurophysiological information. But instead of having all the information in terms of all the activities of all cells, action potentials, and so, we take mean values. So we take a mean value, a so-called mean field of all the synaptic activity, which represents the local EEG, all mean value of firing rates of the cells, which represents then the average firing rate of every cell. And in this way, we did a model where you have two populations of cells, so pyramidal cells and interneurons, interconnected, and in this way you can reproduce activities like alpha rhythm, for instance, or depending on the time constants and depending on the connectivity of the cell of the models, you can also reproduce other kinds of oscillations. So this is a general basic of the neural mass models. I'd like just to show you one application of this by looking at one event that is the so-called event-related desynchronization, event-related synchronization of mu rhythms at the level of the scalp. Now, in relation to an event, the event is a voluntary movement. And that shows you the possibility of using a neurophysiological responsible model, biophysical basic model of neural dynamics to try to understand the puzzle of this EEG, MEG phenomenon. So if you have this movement, what you see here on the horizontal axis is time in seconds, and the moment zero is the moment that the subject makes a finger movement, a very sharp finger movement. You have before that, you have a decrease of mu activity, and the power of mu activity, so alpha range activity in the somatosensory cortex, and this activity 10 to 12 hertz is decreasing. That's why we call it event-related desynchronization, assuming that if the cells are less synchronized, the power is going to be less as well. Then you have, after this movement, you have a rebound of beta activity. So this is increase of beta activity that we call then event-related synchronization. And we have, just before the moment of the movement, you have an increase of gamma activity, which represents the activity of the motor neurons in the cortex, of the motor cortex, just sending the signal for the movement to be effectuated. So this is the phenomenon. Now what is interesting now is that if you ask now the subjects to do movements of two hands, and we plot this now we have to record it, of course, with Gerd Futscheller, this in many different areas covering the cortex, then you see these red areas. Red means event-related desynchronization. So you have the areas of the hand, which are, of course, more lateral to the midline, very clear. Also the supplemental motor area, which is just in front, which always be evaluated by this kind of movements, activated by this kind of movements. That is very simple. But you see also a blue thing in the middle. And this blue thing in the middle is of course a little bit strange, was not expected. Why does activity increase in one particular area of the head, the movement here? And this is event-related synchronization. So that area in the middle is the area that corresponds to the lower limb, to the leg and the foot. So what we did was to do the experiment now by moving the foot. By moving the foot, what you get is a sort of mirror image of what we got with the hand. You see the decrease of activity, event-related synchronization over the hand areas, and you see in the middle, you see now event-related desynchronization. So you have now the opposite situation, which in effect is very consistent in the sense that the area that is being focused attention is desynchronized, and the other areas are increasing the synchronization. Increased synchronization means increase of inhibition, which means that you have, let's say, in this way, a suppression of inputs that could affect the movement that was being, main movement that was being done. Now, the puzzle is this. Now, how can you explain this? How can you get, go over this one, how can you explain this? How can you get some connection between, some crosstalk between the hand and the system and the foot system? How can it be? We know that it is not in the cortex. So where it is not in the cortex, it must be, it's also not in the periphery, so it must be in the thalamus. So this was the hypothesis, and now a neural mass model can be used to test this hypothesis. This neural mass model, I show you here the basic anatomy and physiology. Here you have the yellow are the thalamocortical relay cells. They project to the cortex. The cortex projects back to the thalamo relay cells. This form what I may call a module 1, which is the module of the groups or populations of cells that are related to this particular movement. Okay? So you have this module 1. But you have also, next to it, you have a module 2, which has, of course, the same type of structure. So this is going to be module 2. But how does module 1 and module 2 communicate with each other? That is the question. And this the possibility is, and that is the hypothesis here, is that this happens at the level of the so-called reticular nucleus. And the reticular nucleus is a very thin nucleus of a group of cells that surrounds most of the thalamus, not the most anterior part, but the rest of the thalamus, and they are cells that are GABAergic. They are inhibitory cells. And they do make synapses on each other in series. And this so-called series inhibition is a very typical property of this nucleus. And here you could have communication between the both module 1 and module 2. So we made a neural mass model, which is similar to the dynamic causal models, but now based on exact neurophysiology, not assumed neurophysiology. And here you have the result. And you see in the valve you have the module 1. Below we have module 2. You see the main communication, I mean the main type of anatomical pathways going from thalamocortical relay cells to reticular nucleus cells, and from reticular nucleus cells back to the thalamocortical relay cells. They form this loop, a very important property, this loop. But what is important also, and that is indicated by the red and the blue lines, is that the reticular cells, because through the reticular cells, the synapses, they communicate with each other. So the reticular nucleus cells of module 1 are going to communicate to reticular nucleus cells of module 2. And so what happens now, if you have a so-called attention alerting signal, you have a modulating input, which is mainly activation of cholinergic inputs that project to the whole thalamus, and they cause, and this is now known physiologically precisely, they cause depolarization of the thalamocortical relay cells, and they cause hyperpolarization of the reticular nucleus cells. So what is the consequence of this? If the cells of the reticular nucleus are hyperpolarized, they cannot inhibit the thalamocortical relay cells. So it is, let's say, it's like you are cutting the loop from the reticular nucleus to the thalamocortical relay cells. And in this way, the gain oscillation that is responsible for alpha activity is going to decrease. That's why you put here the blue arrow. And this means that you have event-related desynchronization in the area corresponding to that particular module at the level of the cortex. But what happens now in the neighboring? So again, because you have less inhibition going from this reticular nucleus, because the cells are already inhibited, they cannot inhibit anymore. So you have a decrease of inhibition, in the surrounding of this particular module. And by decreasing inhibition, what you have is an increase of the loop of the oscillation causing the alpha activity, which enhances, and you have an enhanced activity there, and you have event-related synchronization in the neighboring module. And this is what we published before already, in making some models of this already for some time, and this really has been confirmed in different kinds of situations. So what you have now, you have a modulating input coming in, you have in the target modules, you have event-related desynchronization, which is now plotted below the zero line, which is the baseline, and you have, on the other hand, in the neighboring module, event-related synchronization. And also this depends at the distance at which you are from one module to the other module. So this is what the outline was, and I'd like to finish just by some points to think over, and these are the following. I would say, at the moment, give priority to studies of effective connectivity, causal connectivity, over functional connectivity once. That's where I think the main thrust should be. Second, pay special attention to estimates of the error of time. Get methods that can give you information about error of time, because the brain works as a time machine. There is no way out of this, and things that don't happen at the same time everywhere in the brain. Third, there's no lack of methods to estimate causal and effective connectivity at the moment, but from a practical point of view, there's a lack of unbiased analysis of the performance of different methods applied to the same and well-defined set of data. This should stimulate collaborative projects between different centers in this way, a little bit in the way that I started with today, showing what has been done by the group of Wendling in the comparison of these functional methods, but now for the causal methods, let's say. And fourth, in addition, it is necessary to design comprehensive validating studies combining different approaches, and not only regarding methodology, but also experimental levels, especially to combine human studies with scalp EEG, MEG recordings with the electrocorticogram, intracranial recordings. I think that's important to go further to understand exactly what we are doing and seeing in the surface. And last, to strengthen the interplay between experiments and computational modeling, in particular, developing more realistic neural mass models with good basic neurophysiological and biophysical information. Thank you very much for your attention.